Welcome to YouTube channel of Computational Genome Biology.
All right, let's talk about one of the biggest, most mind-bending puzzles in all of science, genome assembly. We're going to dive into how scientists take these billions of tiny, jumbled DNA fragments and, somehow, piece them back together. It's like building the ultimate blueprint for an organism. And trust me, it's part detective work, part pure supercomputing muscle.
So picture this. A DNA sequencer gives you back your data, but it's not a nice, neat book. Nope. It's like someone took the entire book of life, ran it through a shredder, and handed you billions of tiny, overlapping sentence fragments. The big question, the one we're tackling today, is how in the world do you put that story back together from this chaotic mess? And just when you thought that sounded hard enough, get this. It's not even a clean, shredded copy. It's more like a shredded encyclopedia that was already full of millions of typos. We're talking errors introduced by the sequencing process itself. So yeah, this is the massive challenge we call genome assembly.
Okay, so where do you even start with a problem like that? Well, the very first step, and honestly, it's non-negotiable, is cleaning up the data. You just can't build a beautiful, accurate puzzle from messy, dirty pieces. So this raw data usually comes in a specific file type called a FASTQ file. And what's really interesting is that it's not just the DNA sequence. The really crucial part is that every single letter, every A, C, G, and T, comes with its very own quality score. Right, so that quality score has a name. It's called a Phred score, or just a Q score for short. And the idea behind it is actually pretty simple, but it's incredibly powerful. It's basically the sequencing machine telling you how confident it is about each letter it read. So a higher score means a lower chance of a mistake, which makes that bit of data way more trustworthy. You'll often hear scientists talk about a Q30 score. That's kind of the gold standard. So what does that mean in plain English? A Q30 score means there's only a 1 in 1,000 chance that that particular letter is wrong. We're talking 99.9% accuracy. That's the level of quality you want before you even think about starting the puzzle. So before we can even start assembling, we have to put on our editor's hat because the raw data is, it's messy. For example, the quality of the text often gets worse towards the end of a sentence or read. You also find these leftover bits of synthetic DNA called adapters. Think of them like leftover staples from the lab process. And sometimes you even get pages from a totally different book mixed in. That's contamination. So the cleanup process is this really methodical step-by-step edit. First, you digitally snip off those pesky adapter sequences. Next, you trim off the low-quality bits from the ends of the reads. And finally, if any reads have become too short after all that trimming to be useful, you just toss them out. Gotta have good quality pieces. All right, our data is clean, the pieces are high quality. Now we can finally start putting the puzzle together. And it turns out, there are two really different ways to go about this. Okay, so the first strategy is what we call reference-guided mapping. This is the easy mode, kind of. It's like putting together a jigsaw puzzle when you have the picture on the box right in front of you. You use a complete genome that's already been solved as your guide. Then there's the second way, de novo assembly. This is the expert mode. It's like solving that same puzzle with no picture on the box, no guide, nothing. You're building it from scratch. So you might be asking, why would anyone ever choose the harder path? Great question. You go de novo when you have to, when there is no reference map to guide you. This is what you do when you're sequencing a brand new organism for the first time, or when you're looking for huge, novel chunks of DNA that by definition aren't in any of the existing guidebooks. It's where the real discovery happens. Now, for this no-box art puzzle, computers use two main strategies. The first is called Overlap Layout Consensus, or OLC. It's pretty intuitive. It just looks for ways that the entire long DNA reads overlap with each other. This works great if you have nice long reads. The other method is the de Bruijn graph, or DBG. This one's a bit different. It chops up all the reads into even smaller, bite-sized pieces called k-mers, and then it figures out how to connect all those little pieces. It's way more efficient when you're dealing with a gazillion short reads, which is super common. Let's just take a quick peek at that OLC method. It's a really logical, three-step dance. First, the overlap step. The computer finds every possible way the reads overlap. Second, layout. It builds a graph, like a roadmap, to figure out the most likely path through all those overlaps. And finally, consensus. It walks that path and builds the final DNA sequence.
kind of averaging out any little typos it finds along the way. See? Overlap, layout, consensus. So, after all that work, we've got something, a draft genome. But hold on, we can't pop the champagne just yet. Our job isn't over. Now comes maybe the most important part. We have to ask, how good is it? Did we actually solve the puzzle correctly? And that is not a simple question to answer. It's totally possible to have an assembly that looks finished but is actually riddled with mistakes. Huge sections in the wrong order, or maybe big chunks are missing entirely. So we need some solid, objective ways to grade our own work. One of the most common but kind of weird-sounding metrics is called N50. Let's break it down. N50 is all about continuity. Imagine you've assembled your puzzle. Did you end up with a few massive connected sections or 1,000 tiny little two-piece chunks? A higher N50 means your pieces are bigger and more connected. It's a sign that your assembly is less fragmented, which is exactly what you want.
But N50, which measures continuity, is just one part of the final exam. A proper quality check is really like a 4-point inspection. Besides continuity, you've got to check for completeness. Is the whole genome actually there? We can check this by looking for a list of genes we expect to find. Then there's correctness. Are all the pieces joined together in the right order? And finally, you check for contamination, just to make double sure no DNA from other critters has crept into your final result.
And that brings us to the big so what? Why do we go through all this trouble? All this crazy, intense computational work? The cleaning, the assembling, the quality checks. It's not just some abstract computer science problem. It is the absolute key that unlocks real biological discovery. And if there's one thing to take away from all this, it's this. A high-quality, well-assembled genome is the foundation. It's the bedrock. It is the map that you build basically everything else on. Without a good assembly, a huge chunk of modern biology just stops. It can't happen. Because once you have that solid foundation, the world opens up. You can start identifying genetic variations linked to diseases. You can compare genomes between different species to unravel the secrets of evolution. You can discover brand new genes in newly discovered organisms, engineer microbes for biotech. I mean, the list goes on and on. The potential is just massive. The crazy thing is, this whole process, from raw data to finished genome, has gotten so much faster and more accurate in just the last few years. It's incredible. And it leaves us with this one final, really exciting question to think about. Now that we can read the book of life faster than ever before, what huge biological mysteries are we gonna solve next?